introduce Vice Provost Helen Malone, who will make some comments just for everybody's information. I was very impressed. She is a graduate of UC Davis, NYU, and then got her PhD in special education at the University of Texas with a special interest in autism and developmental disabilities. And she's taken a leadership role in that area throughout the course of her career, in addition to being responsible for helping with that program in faculty development here at Ohio State. Dr. Malone.
Well, you don't have to clap, but thank you. Appreciate it. So I think I come, I don't know, Jackie, twice a year. Do I come twice a year? Every time that you all induct a new set of members into the academy, and every time I enjoy coming and visiting the group. I did a quick search as I always do to just remind myself how many members there are and sort of where you're at. And what I saw this year that I hadn't seen previously is when you type Emeritus Academy, the first at least in my Google search, it says Emeritus Academy and then auto loads Ohio State. And then the very next option is Penn State. And if you click on Penn State's Emeritus Academy, it is designed almost identically to what you all have been developing over the last 11 years, I think, if my math is right. And so Penn State now has an Emeritus Academy that also has a lecture program, that also has a grant program, that also has very similar goals to what you all have. And they designed their program after what you all have done. And to me, that's a point of pride from Ohio State to be able to say we have another Big Ten institution that we have this deal in the Big Ten that if you see something you like from another institution, policy-wise, language-wise, you just borrow it. And when you read their website, it's as if it was, they deleted Ohio State, they put in Penn State, they made the colors blue, and it's now sitting at Penn State. And so you all should take pride in the fact that the work that you do as members of the Emeritus Academy was recognized at Penn State and that they've taken that work on. So I thank all of you for that work. And then I think we go to the next thing, which is to induct our new members. And I don't know who's leading that.
Well, we want to congratulate and welcome the following people who I believe are here, and maybe you stand or come up and get your certificate. Clifford Beal from the Department of Biosciences within the College of Dentistry. And Kloos from the School of Health and Rehabilitation Sciences within the College of Medicine. Mario Perugia from the Department of Statistics within the College of Arts and Sciences. Carolyn Wagner from the John Glenn College of Public Health. Maybe she'll on Zoom. Ali Rikabi, who is on Zoom from the Department of Radiology within the College of Medicine. And then we have several people who may be in attendance, and if you are, please come up and get your certificate. Kathy Fagan Grandinetti from the Department of English in the College of Arts and Sciences. Jacqueline Leversridge from the College of Nursing. Barry Lyons from the School of Earth Sciences within the College of Arts and Sciences. And let's see, Marcia Naharikan Nelms from the School of Health and Rehabilitation Sciences within the College of Medicine. Todd Slaughter, Department of Art, College of Arts and Sciences. There were two who notified us that they were unable to attend. Howard Klein from the Department of Management and Human Resources within the Fisher College of Business, and Michael Schwartz from the Department of Near Eastern and South Asian Languages and Cultures in the Colleges of Arts and Sciences. We also have Dr. Louis Clayman from the Division of Periodontology in the College of Dentistry. If he's here, could you stand up so everybody can? And you already received your certificate, I believe. I believe you have your certificate. If yeah, so congratulations to everybody. I think just as the provost said, just looking over the diversity of scholarship that remains within Ohio State University is a matter of pride. It's very well populated with all of the disciplines, and there's very few universities that can match the talent here at Ohio State University. So congratulations to all of you. We also have a lecture today, and Dr. Boyd is going to give us an interesting lecture on effects of drugs on the brain. And he has an interesting, very talented history as well. He got his undergraduate degree at the University of Illinois in physiology and his master's degree in biology from the University of Memphis. And then he got his PhD in molecular biology at the University of Texas, Austin. And his early studies focused on immunoglobulin light chain genes, which is of interest to me. And in addition to that, he has spent his career looking at nicotinic acetylcholine receptors and the potential that they might have for binding drugs, which would have an effect on behavior and character. So today's topic is your brain on drugs. So Dr. Boyd, thank you. Here, use this one.
I got my portable mic. Should be okay, I think. Thank you for that introduction. I'm excited to do this, and I'm also a little nervous. And I'm usually not a nervous speaker if people talk to people that I know. But during my time here in this just a little over a year, I've heard Michelle Herman doing some readings that were really, powerful readings. I've heard Tim Barra talk about the peopling of Polynesia. I heard Barbara Hager talk about the paintings of Rubens and Rembrandt. Dr. Renato talked about robotic art, and I'm not doing it justice by saying that, but if anybody's there for that talk. Dr. Jones talked about Olivetti and the company and the Frank Lloyd Wright influences on the architecture. And so I wasn't really very familiar with any of these when I came here, and I was able to learn a lot. And so I think they've set the bar sort of high, and I'm hoping that I can do something here that has the right blend of science and also allow, something that everybody can take away from this, even if you're not a molecular biologist or a molecular neuroscientist. So my title is going to focus mostly on one drug and then a second drug. And as Dr. Griever said, my work really focused on acetylcholine receptors, which also bind nicotine. And so nicotine would be that drug. And I'm going to talk a lot about that, how it affects the body and why does it affect the body. People, think, well, nicotine, it's from a plant. Why does it do so many things? So I'm going to try to give you the background on that. And at the end, talk a little bit about cannabis products because I think they have some things in common, as you'll see. I mean, the big overlying message is that nicotine affects a normal neurotransmitter system that we evolved a long time ago. We didn't evolve receptors for nicotine. We evolved receptors for acetylcholine. And this has sort of usurped this system, and we've used it for various things, good and bad, mostly bad. And the cannabinoid system is similar. We have an endocannabinoid system, as I'll briefly discuss. And this is a system that's very important for our nervous system function. And again, we've sort of usurped that or taken it over by exposing our body to various cannabinoid substances. And again, some of the effects may be positive, ultimately therapeutically, and there's a number of ones that are probably not, similar to nicotine. So going to focus on what disease affects most of the people in the world. And this is one that I sort of like to talk about. It's probably nicotine addiction. Now, if you think about diseases, there's a lot of cancer. I mean, I haven't added up all the cancer cases, but in a report that came out just a couple months ago by the World Health Organization, we still have about 1.2 billion people in the world that smoke. And if you smoke, you're addicted to nicotine. People will say it's a habit. It's just a habit. It's not really an addiction. No, it's an addiction just like to cocaine or to heroin. In some cases, it's even harder for people to stop nicotine than those. And that actually is a reduction. If you look a few years ago, it's gone down a couple 100 million. So we're moving in the right direction. But again, 1 in 5 adults in the world are still smoking. Now we've got another issue that's starting to come up is vaping. We've got 100 million people estimated to be vaping now. And I'll talk about vaping a little bit. And the idea is, well, vaping is going to be safer, it's better. Probably not. Probably not. It's got its own dangers. Some may even be worse for some people. We've got still a lot of deaths from smoking, about 8 million estimated in the world. At least 500,000 US are directly from smoking. It's no other causes, it's just from smoking. And almost 1 million people have died from exposure each year to just secondhand smoke. And we'll mention that a little bit, that, that was always something, we had to have our smoking-free areas, secondhand smoke. But now we realize that you can be exposed to enough secondhand smoke to get a concentration of nicotine in your blood that's almost comparable to the smokers themselves. And so you are sort of smoking when you're in that room. And there's even a concept now, I'm not going to say anything really about, of third-hand smoke. And this is basically nicotine is sort of an oily, lipid-soluble kind of product. And so it lands on surfaces, and it's oily, and it sort of sticks. And then you have little kids or babies are walking around touching everything possible, and they're putting their hands in their mouths, and they can actually get exposed to this. They're actually seeing some effects of nicotine that's been on surfaces that babies are picking up, and it's actually getting into their bodies. So only, I say, 34 million or so in the United States, but 80% of them start before age 18. And I think that's where the vaping problem comes up that I'm going to mention, is that a lot of vaping starts in that age range. And it's sort of presented by the tobacco companies as a way of saying it's really safer, it's better, it's a way to help you stop smoking. But I don't think that's really been shown to be the case. It's actually showing more and more data saying it's probably a gateway drug that's getting people to use regular cigarettes or vaping products with even more nicotine. And I point out this is a horrible number, but I'm again trying to contrast with what's a major health problem is it's horrible 1 million daily heroin users approximately, but we have way more than that nicotine users. Now, in the past, you may have seen some of these ads. I'm sure nobody here is old enough for that, but the idea is that, look, doctors, they smoke Camels, really good, really cool. Luckies, they're less irritating. It's toasted. This guy looks sort of toasted there too, but maybe on something else. But, this was advertised as being more doctors. There's the woman there. She's examining a patient or something while she's got her cigarette going. Even in the lab. I mean, this violates all sorts of lab protocols, but you've got looking through a microscope and smoking a cigarette. And these are always advertised as, no unpleasant aftertaste, or, menthol cigarettes that have this cooling type flavor. And as I'll point out, menthol is actually an additive that affects addictive properties of nicotine. It makes it actually more addictive. And menthol by itself affects the function of the receptors, even really without nicotine around. So I don't know if they knew this back then, but menthol really makes it even worse. And of course, your baby wants dad to have the best of everything, so he needs to smoke the Marlboros. That's what your kid wants. And then Santa, who doesn't love Santa? He's a smoker too, apparently, but he likes Pall Malls. And I grew up in a household of smokers. My mother and father both smoked, and so I was exposed to lots of secondhand smoke over the time, and I'm lucky I never could do it. Talk about a little bit about how people start smoking, and some people have such an aversive effect at the beginning that can never progress to addiction. But a lot of people don't have that aversive effect, and they can progress to addiction. This is more likely what you see now. I mean, the cigarette companies are pretty sad. The intention of design cigarettes to make them more addictive. Other signs you can see places say it'll kill you, it'll kill the people around you, it'll kill your baby, but people still buy them anyway. Because again, it's one of the most addictive compounds in the world. They even admit now, of course, it causes more death than murders, AIDS, suicide, drugs, car crashes, alcohol combined, is simply cigarettes. And you might say, well, we didn't know all this in the past. Now we've learned. We've learned since the Surgeon General's warning, but I've got a book here that talks a little bit about this. It's an old physiology book. Talks about nicotine. peculiarly common to all of this is that when there's benumbing effects, the real weakness becomes apparent. There's an overwhelming desire for more of the drug to benumb the increased weakness. This enslaved habits are formed. Pure nicotine, 2 or 3 drops will kill a person. When used continuously, the body becomes accustomed, so that it does not produce a great feeling of sickness. Instead of producing weakness, it acts more to benumb the cells. This is sort of their developing tolerance. People have to take more and more of a dose to stay regulated, or to prevent the addictive process or prevent the withdrawal. Says men laugh at the idea of being slaves to such a small thing as smoking or chewing, but when the habit is interrupted, there follows a peculiar unsatisfied and nervous feeling, which few men are able to overcome. Withdrawal. Got some more quotes, but I think you get the idea. These books aren't in very good shape. 1897 is when they were published. So it's not really a new concept. It's been sort of hidden because there's a lot of money in this drug. But people knew that it was addictive, that you develop tolerance. There's several other passages, talks about increasing heart rate, heart disease, lung disease, all in 1897. And to quote Mark Twain, he said, giving up smoking is the easiest thing in the world. I know because I've done it thousands of times. So that's just how it works. So what does it do to your body? And I'm not just going to talk about the brain and addiction, but it does lots of other things too. It produces impacts on hearing and improves vision, gums, the skin, blood pressure. You know, smokers often, they're hardcore smokers. Skin's usually not all that great. Blood pressure, increasing fat deposits, and of course, lung damage. And what I'm going to try to impress upon you is this all happens because nicotine has effects on multiple places in the body. Nicotine acts on these acetylcholine receptors that are very widely distributed. And they're not just in the brain, they're not just in the nervous system, they're on many of our cells in the body. And so when you expose your body to nicotine, you're affecting lots of other things besides just the brain. And that's why you say, how could this one drug have so many horrible effects? Because it has many, many targets. That's why it has many, many targets. It affects estrogen levels, it affects testosterone levels, it affects causing you to produce more fat, less lean muscle mass. It sort of leads to, in some people, type 2 diabetes. It weakens your immune system. I'll show a little bit of data where we showed that these are expressed in immune cells, but people generally, nicotine is an immune system suppressant. People smoke, tend to get sick. Sometimes people smoke, their immune system is suppressed. We have a system that's surveilling the body at all times for maybe some cancer that's sort of early forming. Maybe there's some cells, the immune system would normally find those and kill it. If you're suppressing your immune system all the time, the body might miss those. And I'm talking about the effects in a minute, not just of the smoke, but of just pure nicotine. So we say, well, we could use a patch, it's not so bad, or we use a gum. Many of these effects I'm going to talk about is just the pure nicotine. It's not all the other horrible things that are present in the smoke, but it's nicotine itself. Weaken bones. There's genetic factors that influence whether people become addicted or not to nicotine. So there's been a few genes that are mapped, and there's some others that results are a little questionable, but there seems to be a genetic propensity to become addicted easier. Some people have a genetic propensity, most likely, where you try that first puff or two. I've never been a smoker. I've had a couple celebratory cigars over the years, but that's it. Well, most people smoke. The first time, it's usually unpleasant for almost everyone. I'm not going to ask for a show of hands who's a smoker in here, but maybe some of you have. And there's sort of feel a little bit of a buzz, but you also may feel a little sweaty, heart rate comes up, feel a little nauseated is how a lot of people feel. And if that persists strongly enough, then they won't really progress, probably. They won't continue to smoke. But for many people, that's not the case. It's such a powerfully addictive drug. I'll tell you sort of a horror story about, I'm not going to mention her name, but she was one of the directors of an institute at NIH, studied nicotine and nicotinic receptors her whole life. She's still around, fortunately, but she was a heavy smoker. And what got her to stop smoking is when she got cancer of the mouth and the jaw and had to have her face sort of reconstructed. And that's finally what got her to stop. And this is someone who knew about nicotine, had studied it for a whole career, knew about the mechanisms of action, and still was so addictive that ultimately that's what led to her finally having to stop. And this is a very prominent scientist in the field. I mentioned a few diseases, and I can't really go, I'm not going to go through all of those, but it has signaling of this system that I'll talk about is associated with a number of diseases. And it's either, for example, Parkinson's. It's a little bit inverse relationship. People that smoke tend to have less chance of getting Parkinson's. So the thought was originally, well, they just died before they got Parkinson's because they died of smoking, but it's thought probably not to be the case. There is some inverse effect. Maybe there's some neuroprotection maybe at some point with the right level. But then you've got all the other horrible things that are outweighing that. Schizophrenia, people are like, well, what about schizophrenia? Well, a certain percentage of the population smokes now, and this number's going down and down, but schizophrenics always smoke, almost always smoke. And so there's some issue, and it's been discovered that they have sort of a sensory gating defect as one of their problems. They can't really filter out things. And when they smoke, that sort of mitigates that. They activate a specific kind of receptor I'll mention, and that sort of is, they're self-medicating. I mean, there's some correlation between autism and animal models where they're actually given some nicotine or a drug that activates the system. In animals, it mitigates some of their behaviors that are somewhat like used as a model for autism. Cancer, I don't have cancer there or at the end, but cancer, nicotine doesn't really cause cancer, but it causes cells to continue to divide. It's more like a stimulation. So it's not necessarily causing them to form a tumor cell, but it's causing the cells. I'm not sure if my mic is giving full power here and there and a bunch up. But it's preventing the cell death program that the immune system is trying to put in place to kill a cancer cell. And it acts as a stimulus to cause the cells to divide. And so once they've formed, it just sort of is feeding the multiplication of a tumor and inhibiting the immune system's ability to kill it off. And in Alzheimer's, the key receptors that are involved in this are the acetylcholine receptors I'm going to talk about. And a lot of the neurons that start dying off are ones that respond to acetylcholine or nicotine. Now, how do they do so many things? Well, it's the components of tobacco. We'll show on the next slide, but it's nicotine itself is maybe the most dangerous of them all, and even additives like methanol. Now, if you look at this lab, I see a couple of people that were colleagues, Jack Rahl, Dick Burry. You would certainly, if you had toluene maybe in your lab as a scintillation fluid, or you were doing some kind of counting, it was in the fume hood. Methanol, the fume hood. Even acetic acid was in the fume hood. You know, these were all put into safe areas. You weren't breathing them. Formaldehyde was usually put into a fume hood, you know, where you wouldn't just have it out in the laboratory bench. But yet, these are all in cigarettes. There's 7,000 different chemicals that are in the smoke. So you've got all that going for you. Here's the Marlboro Man. Maybe some of you have seen over the years the Marlboro Man. There he is. He's looking pretty good, although his testosterone levels were lowering. His body fat was coming up because of the effects of the cigarettes. He didn't know at the time. But ultimately, there he is on the right, not looking so good anymore as a result of his smoking. So why does it cause so many problems? Highly addictive, as I've said. Only 7% of the people who quit on their own remain abstinent for 1 year. My wife quit 15 years ago. It was very difficult for her. She tried gum, she tried patches, and finally she was able to get to that point where she could do it. But most people have really difficulty doing it on their own. There's some treatments, there's therapy, there's counseling, there's a couple different drugs, and we'll mention a little bit why those don't really work all that well because of the biology of the way nicotine works in the brain for most people. It's interesting to note that even if you just, as we did in our laboratory, had a bottle of nicotine or a powdered form of nicotine, that it's considered to be an EPA hazardous waste. It has to be disposed of as a hazardous waste, not sucked into your body. Again, 60% of smokers die of a tobacco-related disease eventually, and it stays in your body for a while. Half-life is a couple hours, meaning that after 2 hours, half of it's gone, another 2 hours, you're down to like a quarter, but it stays around. And one thing that makes it difficult sometimes to study in animals is in mice, it's only a few minutes. And so you've got to figure out how do we dose the mice and get an equivalent situation to we have in humans, and that makes sometimes the studies a little difficult. It's a very effective drug delivery system, or cigarettes, reaches the brain in 10 seconds. Why does it affect the nervous system? Well, because it acts on, as I said in the intro, an endogenous system of a neurotransmitter that we use, acetylcholine. There's nicotine. The structures don't really look, you think, well, I don't know how those look so close together. But if you look at up in the upper left, this represents the structure of the receptor, sort of at a molecular kind of model. You see the red blob is nicotine. It fits right into a nice little pocket. And that pocket where it fits, as we see, is the same pocket that acetylcholine fits in. So our normal signaling process, there's a pocket, acetylcholine sits in the pocket, nicotine sits in the same pocket, and other drugs do as well. So again, sort of taking over the signaling. And the simplest take-home message, if you don't get anything out of it, is that nicotine is stimulating some cells when they're not supposed to be stimulating. It can also inhibit cells. So it's inhibiting cells when they're not supposed to be inhibiting. So you're basically disrupting the normal signaling process with this exogenous drug. One thing to point out here, these acetylcholine receptors I'm going to talk about in detail for a few minutes are present in the brain, like I said. They're also present at your skeletal muscle, all of your neuromuscular junctions. So maybe you've heard of this before. Somebody gets bit by cobra venom. That's cobra venom. It goes into the pocket a little different way, but it basically takes over the whole thing. It binds very tightly. Then it basically paralyzes the muscles. And so once you put this on the pocket, the normal neurotransmitter can't get in there. There's no more signaling, and people die of a paralysis very quickly. Again, not to go too molecular, but I think everybody can sort of see the idea. This is what the receptor looks like. And we're looking from the top down. This is the one from the muscle, but it's got 1, 2, 3, 4, 5 proteins that make it up. So it's like a barrel stave, sometimes people say. And down the middle, there's an opening. And if you look at it from the side, it looks a little crazy, but this part at the top of the screen would be outside the cell, in the synapse, or outside of a skin cell or a lung cell or a pancreatic cell. We did some work to show some of the genes are expressed in pancreas and in pancreatic tumors. And then this gray line represents the membrane, so it goes through the membrane inside the cell, and it forms sort of this channel. Here's a version of another one. This is one that's expressed in the nervous system. You can see it's basically the same overall 3D structure. They're very similar, and they work in a very similar way. Now, the one at the muscle has a pocket, and you can see the little gold, maybe. This represents where acetylcholine would bind, or nicotine would bind. And there's 2 pockets. It's sort of like a old movie where they have the nuclear launch codes, and you got to have the 2 guys with the keys turning the keys at the same time. You really have 2 acetylcholines will bind, and it's going to open up this channel and stimulate the cell, or 2 nicotines. It's the same kind of story here. 5 different subunits, but they're going to be different proteins, but they're very similar. The muscle one is not really as sensitive to nicotine as the ones in your brain and elsewhere. It doesn't respond very well to nicotine, which is good and bad. If they responded to nicotine, nobody would have ever smoked because everybody would have been paralyzed and dead when they first tried nicotine. So it doesn't really, we can smoke, it doesn't affect our neuromuscular receptors, but it affects obviously our neural receptors. Although they're very similar in overall 3D structure and shape. They've got 5 subunits, they've got sort of a little channel down the middle. This is a cool picture and done by some work by a guy that's done some amazing microscopy. But the idea is acetylcholine or nicotine will open the channel when it binds in that little pocket. And on the left, this channel is closed. It looks like there's some space, but it's not enough space to let through what is normally going through. And then when nicotine binds, the whole thing does a little bit of a shape shift, opens up the channel. When that channel gets open, what happens to the cell? Ions flow into the cell, in this case, sodium. So outside of our cells are pretty high in sodium compared to the inside. We open up the channel, sodium comes in, it's got a positive charge, Cell gets positively charged, and you could say it's just simply exciting the cell. It's stimulating the cell. And if it's a neuron, it's going to send its action potential, its electrical signal. It's going to release its neurotransmitter in response to being excited. So again, when you can see we've got the pockets here too, and they're a little ways down from the top. You've got two acetylcholines, or you have your nicotines binding, a little bit of a shape change. This opens up, sodium comes in, the cell gets excited. And so this happens with acetylcholine. It's also going to happen with nicotine as well. Nicotine gets a little more complicated in that nicotine also has a propensity to turn off the receptors, or to so-called desensitize them. So nicotine has a really complex mechanisms of action: excitement, then desensitization, excitement, then turning off. And so again, that makes them a little difficult to study, to understand. There's a little channel that opens up, sodium comes through. Some of them have calcium go through, that sends a completely different signal, which is another story. Not doing neuroanatomy. I'm not a neuroanatomist. Dick's probably more of a neuroanatomist than I, but not really. We have some people in our neuroscience department that are. But I'm showing you just to give you the general idea. How can nicotine affect so many different things I'm going to talk about? Because these red arrows in the blue and the yellow, these basically represent areas of the brain where there's a bunch of acetylcholine that's released. So acetylcholine is released over wide areas of the brain. This is our cortex. It's released in the thalamus. You think of the thalamus as sort of a grand central station in a way. Lots of information coming up, information coming down. So it's fine-tuning. It's involved in focusing attention, areas of the brain where we have learning and memory. We've got pathways that are involved in addiction that we'll point out. It's expressed many places. It's got in the vestibular system, sort of involved in balance and coordination. These little guys down in yellow are motor neurons in the spine. Bottom line is it's all over the place, not just the neuromuscular junction. If we look at the sympathetic nervous system, again, don't worry about the details. Just look at the little green dots in the blue. Acetylcholine is released from our autonomic nervous system, and everybody who's experienced all the various effects of the autonomic nervous system, maybe not thinking about it so much, but we've got the sympathetic nervous system, sort of maybe called, you've heard the fight or flight reflex. You get excited, focused, heart rate goes up, you're scared, something happens, you focus you. Versus the parasympathetic is sometimes called the rest and digest part. This is when you're digesting food and calming your heart rate down. And acetylcholine, again, is the normal neurotransmitter for both some of these sympathetic nerves and the parasympathetic nerves, and they work on these receptors that also bind nicotine. Now, if you're looking at this closely, you'll see the other side of the story that I'm not going to talk about. What's this guy down here that also binds acetylcholine? This is a whole nother set of receptors, and they're called muscarinic. And so if you're wondering about those, those are a different kind. They're not a channel. Nicotine does not affect those. So that's why we're sort of putting those away for today. If you look at this, again, sort of a crazy little figure, but the bottom line is everywhere you see these blue lines, these are releasing acetylcholine. And so if you look over here, you've got sympathetic. They affect pupillary dilation, salivation, heart rate, lung, pancreatic function, digestive tract function, bladder function, and so on. These are all being controlled in part by acetylcholine, acting through acetylcholine nicotinic receptors. Okay. Any questions so far about that? I mean, that's just, again, trying to give you not make it a challenging test, but to say so much diversity. And this is where the diversity really comes in as to the type of receptors. Even though they're all receptors for acetylcholine, and they're all receptors for nicotine, in the nervous system, we even have more variety. If we went back to the neuromuscular one, we have the 5 different proteins, and they were given Greek letter names: alphas, beta, gamma, delta, epsilon, just to denote them. But when people started working in the nervous system, and we started looking at them in an animal we started studying, zebrafish, many people were able to find there's actually 9 more that are like the one in the neuromuscular junction. alpha 1, the neuromuscular junction. They just started naming them alpha 1, alpha 2, alpha 3, alpha 4, and so on, all the way up to 10. So there's 10 different kinds of these proteins expressed in the brain. And then they started looking around and found a few more that are betas, they call them, that are not like alpha, but they're also part of the receptor. So the theme is still the same. 5 proteins, channel down the middle. The little black triangles represent where nicotine or acetylcholine will bind. But if you do the math, adding up to 5 with this many alphas and betas, you could have 10,000 different kinds of receptors. We don't have that many, but we've probably found at least 20. And many animals have the same set of genes. We studied zebrafish to the end of my time. We cloned 9 different nicotinic receptor genes from zebrafish, and we're able to compare them to rat, mouse, human, almost identical. The protein level sequences are almost identical. And so it's part of the same big family. But this produces some great flexibility for the nervous system. When you have that slide where the red arrows represent nicotine being dumped out onto various, or acetylcholine dumped out, involved in memory and motor functions and focusing and attention and learning and all that. At the end of the line, though, the receptors, even though they are now called nicotinic acetylcholine receptors, there are different subtypes. They have different combinations of subunits adding up to 5. And what that means is, even though nicotine or acetylcholine will open them, they respond slightly differently. Some open a little slower, some open faster, some close really quick after they've been opened and no more sodium, or some stay open for really long periods of time. Some that have a really high affinity for nicotine, meaning just a little bit of nicotine, will activate it. Some you need a lot more. Sometimes some you need a medium dose. So this is where it gets complicated in trying to understand all the effects of nicotine on the body, because they're taking over a system that's very fine-tuned with lots of different types, lots of different locations, and now you're just throwing in an exogenous drug that's sending a signal when you decide you want to do that signal. And again, you're throwing a monkey wrench into the normal system. So this just represents one example of one of the neural ones. And again, you see you've got the same 2 alpha 4s, 3 beta 2s. To make it even more complicated for people, oh, suppose you have 3 alpha 4s and only 2 beta 2s. Still at 5. That's totally different function. It has a completely different function and different sensitivity to nicotine. So it makes it a really difficult problem. And part of this is why haven't we developed drugs to help people stop smoking yet? Because it's hard to find the right targets. Because acetylcholine receptors are involved in so many things, and they're so similar in their overall 3D shape and the pocket that, yes, you could find some drugs that would block this signaling, of course, but how much damage would you do? I mean, you disrupt signaling in all sorts of different systems. It can't just be a blunt force instrument like we're going to block all this cholinergic signaling. Again, crazy looking figure. I'm violating rules. I tell my students, don't put too much stuff on a slide. I'll violate this a couple times. But again, not to be the details, here's a human brain, monkey brain, and these letters represent different types of acetylcholine receptors. So even in just certain little areas, basal forebrain, really important for learning, focusing, lots of function, multiple types. You look at this area here, we'll talk about the addictive properties in a minute. Ventral tegmental area, this is where dopamine is being released into midbrain. Tons of acetylcholine receptors of different types. Hippocampus, learning, spatial learning, all sorts of different learning. The bottom line is, all over the brain, you have these different types. And these different subtypes actually have slightly different functions, even though they bind acetylcholine and open, they bind nicotine and open, they do it in a little bit different way. So again, it makes a very complex problem. Now, I use this slide because probably nobody, maybe people remember the old movie, Good, Bad, and the Ugly. It's not really good, but what are some of the effects of signaling through these neuronal nicotinic receptors? Well, they affect cognitive enhancement. So in some cases, nicotine does. I mean, people smoke, perks them up. They get a little bit of a buzz. They're a little more focused for a while. Anxiolytic, relaxing. Again, it produces some arousal. Antidepressant activity. So again, there's reasons like, why do people smoke? Well, they're addictive, they can't stop, but why do they do it at the beginning? Because there are some effects that people get from the cigarette that seem to be positive. And these are things that are also regulated by the normal nicotinic receptor system. You know, it's involved in enhancing cognition and anxiolytic, you know, relaxing, things like that. So these are all things that are done as part of the normal signal. And then people say, well, I don't have enough of that signal, and maybe then use nicotine to enhance it. Analgesia, there's some pain, anti-pain actions of cholinergic systems. But I put the bad and the ugly from the old Clint Eastwood movie. Again, we've got a role in Alzheimer's disease, Parkinson's disease, schizophrenia. Again, all the signaling pathways are not fully understood. There's some genetic linkages between some of these diseases and genes that encode these receptors. Tourette syndrome, some epilepsies are genetically encoded only because of a mutation in one of the acetylcholine receptors. As I mentioned, cancer growth promotion, of course, addiction. And this drug works very fast, and it works on a main population, and this is the one that we tried to target, others have tried to target for smoking cessation. And if you've heard of Chantix or varenicline that was used for smoking cessation, it's targeting this type of subtype of the acetylcholine receptors, the alpha-4 beta-2s. You can see what happens. I mean, the idea is here, they've labeled all those receptors. So you can see there's heavy levels of red and yellow. One puff of a cigarette, what's happening is the nicotine is coming in and bumping off the normal neurotransmitter. So you're basically getting rid of these areas that have a labeled tracer that recognizes these receptors. 3 puffs, a cigarette, 3 cigarettes, you've pretty much taken over all of this high affinity type of acetylcholine receptor has already been dysregulated, basically, or controlled by nicotine. And the sort of a dilemma you might say is, okay, why do people smoke all day if after 3 cigarettes you basically sopped up all the signaling? You know, you've sort of taken over all this signaling pathways. And the answer is, in part, is that there are other receptors for nicotine, some other acetylcholine receptors that do not have that high affinity. It takes a little more. So it's probably working on some of the other ones too. But we know what some of these receptor types are. And we know that some are involved in the addictive properties of nicotine, some of these receptors and some of the pathways, and some of them are involved in more what we would call the aversive effects of nicotine. And these have been studied, and so this is a good area of research for future. We know that people smoke to a certain point, and then they titrate how much nicotine they have. You know, they say, I've got this much, I'll stop, or I'll have another cigarette, or I'll wait for a while. You think they're planning, but your brain is basically telling someone who's smoking, you need a little more, you need a little less, titrating to the right level. And it's to maintain this reinforcing addictive effect of releasing dopamine, but preventing the aversive effects when you have too much. And they've done this in mice where they know which acetylcholine receptors are involved in the aversive effect, and they can genetically knock those out. And this poor mouse just keeps administering nicotine, 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 nicotine. It doesn't have the aversive effects. Its brain doesn't tell it to stop. And yes, mice will self-administer nicotine. That was always sort of a mark of addiction. People said, well, cocaine, they self-administer. And opiate drugs, meth, lots of things like this, these animals will self-administer. But no, they don't self-administer nicotine because nicotine is not really addictive like those other drugs. No, it's addictive exactly like those other drugs. They will self-administer. And the idea of doing this is that it stimulates dopamine release. All these drugs sort of feed into the same pathway. When you do something fun and healthy, do your hobby, you're releasing dopamine in certain midbrain regions. Sex, gambling, overeating, that's sort of what I like to do. This is releasing dopamine in these areas. Cocaine, heroin, nicotine is also stimulating these dopaminergic systems. I mean, we like that, obviously. And nicotine is very efficient at stimulating the release of dopamine in these addictive pathways. These acetylcholine receptors are there, very complex signaling that's normally taking place to keep the right amount of dopamine present here. And then you dump in nicotine, you get this surge of dopamine, And now we like to keep it up. We develop a dependence and tolerance very quickly. The rewarding and aversive effects that are sort of balanced out by our brain, where it tells us to smoke another one, stop, that's good, and so on. Abstinence, of course, produces these same symptoms you have when people try to withdraw from heroin or cocaine or something else. Stress, anxiety, irritability, headache, low heart rate, blood pressure, and so on. My mother, heavy-duty smoker, highly addicted to tobacco. Little old lady, though, very calm. When I lived in San Diego, I was a postdoc out at UC San Diego, and she flew from Minnesota out to visit us for a month, and she stayed with us. And during that time, Northwest Airlines decided no more smoking on the plane. She was almost one of these people you see on videos now where they had to practically put her in a straitjacket and tie her up to her seat. She was going so berserk because she didn't have her cigarettes for those 4 or 5 hour flight that she practically had to be restrained. And so this is how powerful this was, this addiction. And she started going into all of her withdrawal symptoms already. Again, this is just a simple one to point out where's these pathways that are important. Lots of different dopamine pathways, but the one we're really going to focus is here. This pathway here that I'm pointing out, that's where there's tons of acetylcholine receptors. That's where the dopamine release is involved in our addictive pathways. And this is, again, sort of a crazy figure, but here's our dopamine neuron. Here's acetylcholine. It's being dumped onto that neuron. Just replace that with nicotine. I mean, there's obviously a normal amount of signaling that we have, but you replace this with nicotine, and we stimulate more and more dopamine release in those midbrain areas that we like to have dopamine release. And if we have a long-term chronic exposure to this, we sort of change these circuits now, as it does with any other drug of addiction. These just show you lots of different receptor types are here. Again, that makes it like really complicated to target one specifically that will help to block the addictive properties and help people to withdraw versus disrupting all sorts of normal signaling pathways. So what happens with chronic use, though, is you actually change your brain, just like you do with other drugs, or just like you do when you do positive activities, right? You learn something, you change the structure of your brain, right? You do motor activities, you become really good at a sport or running or jumping, you really restructure the motor parts of your brain, you train. And you do it here too. Ends up you upregulate the number of receptors. So now the cells are on the surface even more. Now they're really sensitive to it. They're ready to go. You actually get higher levels of this dopamine release, and now you need to maintain that higher level. Your brain is adapted structurally, signaling-wise. You have to maintain it. Strangely, what's been discovered a few years ago by Henry Lester at Caltech, who's done amazing work in this field, that nicotine seems to just stay in the body for a while too. Synaptic vesicles are where the neurotransmitter is sitting. So a cell that's releasing acetylcholine or dopamine, it's got it in a vesicle. The nicotine seems to be able to get in these vesicles and just hang out for a while. And then when that vesicle is released sometime later, it's also released. So you get this continuous, you still get nicotine being put out signaling even after smoking is stopped and the nicotine's been cleared from the rest of the body. And as I mentioned, additives such as menthol really increase the addictive potential because they increase the number of receptors on the surface and they regulate their function. And I want to give a plug for a student here who graduated from Dennis McKay's lab in the College of Pharmacy here. Brandon Henderson, who's now professor at Marshall at the medical school, has done some amazing stuff here since he left here on really understanding the role of methanol in these trafficking and addictive properties and so on. So I'm giving him a shout out as an OSU grad who's doing really good work in his own lab. And the last thing is even to mention, if it's not complicated enough already, what we call epigenetic changes. And so what this means essentially is that you're not changing the DNA sequence, but you're changing the regulation mechanisms that occur at the chromosome level. So nicotine seems to actually change the way the genome is regulated after some long-term exposure. They can see clear changes in some of these epigenetic markers, and then they look downstream, they can see changes in certain RNAs, proteins, genes that are expressed. In other words, it's changing for a long period of time the cells that have been exposed to the nicotine. And I keep pointing out with acetylcholine, but remember acetylcholine receptors are on other neurons. They don't just release acetylcholine. They receive a signal, they open their channel, cell gets excited, dumps out its neurotransmitter. One neuron is thought to maybe communicate to as many as 10,000 others. But that cell that has acetylcholine receptors can dump out dopamine, norepinephrine, glutamate, and you see all these things that they're involved in. So nicotine is not just affecting acetylcholine, it's affecting dopamine, as we've already learned. That's why it's addictive, but also it affects all these other systems as well. It's amazing that actually anybody who smokes can actually survive in a way. It seems to not go insane. Where are they expressed? I showed at the beginning when people have bad skin, we have vision problems, we have heart problems, and so on. Receptors have been identified in skin, leukocytes, kidney, bladder, lungs, reproductive system, heart. And when they look at these, when they look at the genes, they're the same ones you see in the nervous system. The same alphas and betas that form these subtypes that I showed you a few slides ago. Those are the same ones that are in these cells. Same sequences, same proteins. And so you might say, well, these guys, they don't really have electrical activity, do they? They don't release neurotransmitters. So what are they doing there? Well, they're still sending a signal that excites the cell. And the thought is that these signals that come through here, maybe it's calcium, something else, still sends a signal to the cell. Doesn't send a same kind of signal it would send in a neuron. And one example of this, some work we did with Mohan Sapori a long time ago at New Mexico. And what we showed is that T cells express one of these nicotinic receptor types. An immune cell, very important immune cell. And this was sort of not really expected. We thought, I got some T cells and I purified this, and yes, it's the same protein, it's the same RNA, it's the same sequence that's in neurons that's in these T cells. But what's different about it is that this one doesn't form a channel. It interacts with other proteins that send a different signal in the immune cell, but it's still in the immune cell, and that makes these immune cells sensitive to nicotine. As I said, nicotine's a big immune suppressant. And then we looked at a different type of cell, mast cells. These have nicotinic receptors as well. And it's different subtypes, but it's the same genes you see in neurons. Again, I got them out, we sequenced them, we looked at them, they're the same thing. And so how does it affect all these systems? It's in many cells. We don't know exactly how it's signaling, but it's present. Now, this baby may not be smoking a nicotine cigarette. It's not sure. From his hat, it might be something different. But this is sort of a disturbing aspect of nicotine, is its effects on development. Now, happily, I've changed this number. It used to be 12%, it's down to 5%, so a lot better. Still 5% of pregnant women smoke. And enough nicotine can get into the fetus, even higher levels than are found in the woman's blood. This is from an old study we did, but it points out that what does nicotine do in embryonic development that may not do the same thing in an adult? And this was using a zebrafish. If you look at the left, we have a zebrafish. It's like it's swimming toward you, and you chopped it right down the middle of its head. And we've got the two eyes, and then we've got a large part of its brain in the middle. So we took these zebrafish, and the reason we wanted to use zebrafish for studying nicotine, there's been tremendous work in rats and mice and primates, but we thought, here's a system we don't have to worry about some of the effects of the other components of smoke or the effects of hypoxia on a fetus. Because if you give a nicotine to a placental animal, you do disturb the blood flow, you do disturb the blood flow to the placenta, to the fetus. So if you see bad things happening, which that also causes bad things, obviously, is it really due to the nicotine, or is it due to all the other lack of blood flow and oxygen? So we were able to say, well, here, you just put in nicotine into the water where the fish are living. And here, untreated, 96 hours is after it's been fertilized, only 4 days. It's already got a brain, swimming around, it can learn. They develop very quickly. Here's where we added some nicotine to the water. And we use an assay that basically shows you when a cell dies, you get sort of a dark spot. And you can see we have quite a bit of death here. And then what we did to show that it was really the nicotine that was doing it, acting through the receptors, is we added another drug here, which basically would block the action of nicotine. An antagonist that would prevent it from getting to the pocket and sending the signal. And we can see we cleaned up or prevented most of that death. So human and animal studies have shown long-term effects on nervous system structure and function, not just in zebrafish, as I'll show, but in humans as well. So another danger of smoking is that you could be disturbing normal development. That acetylcholine is not just functioning in a mature brain, it's not just a functioning as part of the addictive process, but it's functioning somehow sending signals during the development of a fetus. This is maybe it's a little obscure to look at, but what we looked at with zebrafish was to say, if it's doing something during development, are these messenger RNAs, and even we looked at proteins in very early embryos, are they actually there? And this is actually looking at maternal RNA. Maternal RNA is RNA that's present in an egg before it's even fertilized. And it's thought that maternal RNAs serve a purpose before the zygote is formed, before the egg's fertilized. We get a zygote and it starts producing its proteins and dividing and, developing into eventually a human or an animal. There's some maternal RNAs that seem to be coming along inside the egg that help it get started. Some of these early processes, they get translated. And so we actually found that 3 of our acetylcholine receptor subunits are actually present even in maternal RNA, which is very surprising. This is in a zebrafish. They don't like it when you do this, but you can squeeze them a little bit and you get some eggs that come out that are unfertilized. And then we looked at the RNAs there using PCR, and we used 72 hours as sort of a control to show we know some of them are there. This one's not, that one's not, but we find that several of them are. So it's sort of a mystery is why are they there so early in development? Now this is when we looked at the one here, alpha-2, one of the subunits. And this is one that's always been sort of a mystery in adults because you really don't find much. A little bit in rats and humans, but not so much. So I thought, what's its role? Well, we think it may be more developmental because if we look at this is a zebrafish. It's not been cut, it's not been sliced. One of the advantages of zebrafish is if you treat them properly with the right chemical, you can keep them transparent. They don't develop melanocytes. They just sort of stay clear. And this big blob is the yolk cell. They have a yolk cell that they sort of feed off of for a while. So the details are a little smudgy here, but we can see even at 10 hours, you've got purple stain that represents cells that are expressing this alpha-2 that we saw in the maternal. And here we are, not much in the brain. This is the brain. We see down the spinal cord, we see a number of areas where it's being expressed during the spine. This one with the lights on doesn't show, but we're sort of looking down its back. We're above it, looking down at its spine. You can see it. But as the ages go on, you start seeing less and less. And by about 4 days, you don't see a whole lot anymore. And so we think that part of a developmental role is transient. It's expressed at a time, serves some kind of purpose, sends a signal. We don't even necessarily have synapses or any kind of communication, but we have some kind of signal that's sent. It's another example of showing you that maybe alpha-2 is important in development, and that here we used a genetic technique to sort of knock it out from a developing zebrafish embryo. And here, these green represents motor neurons in the spine. So we've used a zebrafish line that's been engineered to show green in these motor neurons. And so here's normal, 72 hours. As we get on a little bit farther, you can see these are axons that are coming out. They look like they're sort of stopping, but they're starting to innervate muscle there as part of their normal process. Now we find some, these are the way my graduate students classified some. Some of them, though, seem to have some issues. This one still can swim, looks pretty normal, so maybe we didn't really knock out the alpha-2 much. You still see the processes. But now we saw 3 other kinds that she's categorized that seem to either have a problem swimming or they had a weird shape. They sort of had a curved spine now. And you can see that as we go along, you're not getting the same processes aren't coming out anymore. Here they're coming out, and normally they may come out down and then go up. They may come out below and come up. But they just sort of come out and stop. At the later stages, they come out, but there's not very many. And even in the most severely affected ones, they don't really seem to come out at all. And those are the ones that can't really swim or do anything. So again, we point out that maybe this acetylcholine receptor has some developmental role. It's expressed for a while, and then it goes away. That's what it did. Here's a study from a colleague of mine it really is interesting too, is using nicotine directly. There we were just looking at a receptor. And this is motor neuron development, so it's sort of the same kind of picture that I showed you before. And this is a 42-hour, 66-hour. They're looking at as motor neurons are starting to develop and form in the spinal cord of a zebrafish. And so if they expose them to nicotine, starting at 22 hours after they've been fertilized, and then you see, they're not so much yet. They look about the same number. We notice that 66 hours, there's a lot less. And the question was sort of, and you can see when the fluorescent version, quite a few less now after 66 hours when they got exposed to nicotine. So what happened? Did they die? Well, we saw in some cases they might die in the brain. Did they die or something else happened? And what they asked the question is said, okay, if we put it in there for 22 hours to 66 of the day, then take it away and let them continue to be 5 days old, what happened? Well, it basically recovered. You didn't kill them, but by exposing them to nicotine, you delayed their normal development. And if you took it out at the right time, they still basically, you see, got about the right number of motor neurons still. They came back, but they do still have some deficits. If you look closely at some of their axons, these guys still have a permanent swimming problem. It's not horrible, but they still have a permanent problem. I mean, this illustrates again the concept that when you expose an animal to nicotine, adolescent, brain still developing, fetal, that you do produce some long-term changes in their nervous system structure. If you needed more concern about the dangers of nicotine, there's another one. As I said, fetal nicotine levels can accumulate higher than in the mother. And we know a lot about some of the obvious things with nicotine, like a low birth weight, and some of the deficits that we're going to describe here briefly and that have been seen are not like a fetal alcohol syndrome where it's really horrible phenotypes, but there's been some comparison, and there was a lot of discussion over the time saying we've got cocaine babies, mothers were addicted, baby had cocaine during development, versus nicotine babies. And then they said, what do they look like when they're 15? And it really showed the cocaine babies, for the most part, have recovered. They seem to be pretty good. Nicotine babies still have issues because there seems to be long-term changes in gene expression. They have sort of a tendency for more novelty seeking, and this is in animals. In humans, it's been shown, and these studies have gone back and forth, but I think they finally settled on this as real, is that boys tend to be 4 times more likely to have ADHD if their mother smoked in utero. And I would be an N of 1 on that case because I've been accused of having ADHD and my mother smoked like a chimney. Girls have 10 times more likely to use drugs of abuse if their mother smoked. And some of those studies I think are pretty solid. And so it would make sense that if you're exposing the developing brain to these, you could produce some kinds of long-term changes. And there's been studies that have shown that there are long-term changes in longitudinal studies of brain structure. Adolescent, of course, is a very bad time. We've been talking mostly about developmental fetal exposure, but adolescence is the time when we said lots of people start smoking or they're starting to experiment with smoking. And during this time, it's very important. And again, you disrupt the signaling. You disrupt the signaling of the dopamine system, you disrupt the signaling of those other systems. And there's some evidence that, again, kids that are using cigarettes during adolescent time have deficits in working memories that persist and cognitive tasks that persist, some brain structural changes that may persist, and they may sensitize them to future use of nicotine or other drugs of abuse. So just a little touching on, like, if you didn't think nicotine was bad enough, and you can see I'm up on my soapbox a little bit, again, I'm trying to get the take-home message for everybody is that it does these things because it works through a normal system. The normal system is very complicated. And when you disrupt that normal system by throwing in a signal that's not supposed to be there at that time, or turning off receptors because nicotine can desensitize them at a time, either way, you're disturbing the normal pattern and normal building of the nervous system. But what about vaping? Is this any better? And the bottom line answer is no, it's not. It's not any better. It's been sold as being better, but again, you've got tons, again, too many words on this slide, but it's got damage as you'd expect because you're burning a lot of chemicals. They have very high levels of tobacco or nicotine, and they even have higher levels in many cases than they actually say they have. They don't advertise properly. It tends to be more slowly absorbed than if you're smoking a cigarette, but you can get to very high levels. The concentration in Juul, which is one of the products a lot of people use, is even higher than in cigarettes. It's even more nicotine being delivered than you get from a cigarette. And the problem with vaping is you get this increase that's occurring amongst high school students. 10% of secondary students say, at least, that they have vaped nicotine. And the age of onset of vaping now is very young. And so I think, I'm up on my soapbox a little bit. A couple million middle schoolers use e-products, e-cigarettes. And the idea is, well, the cigarette companies, less and less people are smoking regular nicotine cigarettes. We've seen that come down. So how do we get more people addicted to our product? Let's put out their vaping. Again, this is an editorial now. They put out the vaping products and say they're going to be safer, it's going to be better, but they're not. They're actually could be just as dangerous or more dangerous. They still produce secondhand smoke. Students who do vape want to quit. Why? Because they're already addicted to the nicotine in the vaping products. And another danger is that people now are starting to use marijuana in these e-devices. An even greater danger is that they're using nicotine and marijuana together in these devices. And I'll give a little bit at the end talking about marijuana during adolescent development, and you're going to see the same story we've sort of seen with nicotine. It disturbs development. Now, when you're older, I've heard a colleague of mine, maybe some of you know, Gary Wenk, he would say maybe as an adult, smoke a little pot, something like that. It may be actually anti-inflammatory, may be good for you. But in development, no. And nicotine is the same way. And nicotine and marijuana is a real double whammy. We've got, I used to review grants for the California Tobacco Research Foundation. And the last time we did this last year, they're finding lots and lots of grants are being proposed because now they find in California, a lot of people are using, high school students using nicotine and marijuana together in vaping products, which is really a dangerous double whammy. We put in a lot of additives, and these additives are supposed to mask the taste and make it better. But these additives themselves are very dangerous. Menthol's an additive. Green apple's an additive. There's other ones. Chocolate chip cookies, cinnamon, things like that. And a lot of these have a warning that's not really accurate. They don't indicate that they contain nicotine even, or how much they have is a lot less. Again, we still have all the harmful products. Could serve as a gateway or help stop smoking. That's sort of the argument people have been having. But it looks like latest literature, very recent, is saying, no, it's not really being effective as a cessation aid. As a matter of fact, it's causing people to move on to traditional cigarettes. It's not really as sensational. And again, some of them have lower levels of toxicants than conventional cigarettes. I guess you could say that's a plus. It doesn't have 7,000 horrible chemicals. It may only have 100 or something. But I don't think that's much of a plus. And again, little is known about the developmental effects of vaping, but there have been some studies that in mice, that when they expose them to adolescent, and you can actually put mice in little vaping chambers and you can sort of recreate the vaping atmosphere, they have changes in their gene expression already. They've changed basically the function of their nervous system by being exposed to this. And even with flavorants, that's the crazy part about it, is that even with just the flavorants, especially green apple, and that's another shout out to Brandon Henderson, who graduated from here. He's shown that green apple can do all sorts of things to signaling in this nicotinic system without even having any nicotine around. Again, this just shows you that these products are the same kind of things you would see with regular cigarettes. And the temperature that's used in these vaping pods can actually be pretty hot. And when you do that, you take some chemicals that they say, well, this is a safe chemical. But of course, when you heat it up, it converts to another chemical. It's actually more dangerous. And so that's why the pods themselves that heat up these are bad. Again, little regulation of the additives. These have been engineered to really deliver high doses now. So I think the people that use them don't realize, and it's not well labeled in the way they function, they actually get way more nicotine than they think they're getting. So they think, well, I'm not getting as much nicotine. This is better than a regular cigarette. In reality, they may be actually getting more nicotine than they would for a regular cigarette. And there's quite a few adolescents who are using it. There's a large increase in high school student use. And again, I pointed out green apple alone can increase nicotine self-administration, just the green apple. And now we're putting green apple in a vape with nicotine because it makes it, I guess it makes it taste or smell better. And then menthol, as I've mentioned. Now, last little part, I talk about cannabis. And I'm going to mention this because it's a similar kind of problem. I'm not saying we should prevent cannabis products. I'm not giving any editorials on any of that, but the cat's out of the bag. But again, we need to think about the science of what's really happening to the brain when you use cannabis products. And the similar system has evolved. Okay, we didn't evolve a receptor for cannabis. People have been using cannabis products for hundreds of years, really thousands of years, thought. But we didn't evolve a receptor. Just like we didn't evolve a receptor for nicotine, we evolved a receptor for acetylcholine. Nicotine happens to bind in the pocket. Same thing here. People have been using cannabis products, and here's the structure of THC. People said, well, we've got to have our own endogenous cannabinoid neurotransmitters. Same what they did with opiates. People used opiates. Solomon Snyder and his group, Candace Pert, like, we've got to have our own opiate receptors, right? Somewhere. And then they discovered we have opiate receptors. Same thing here. We use cannabis products. Well, we must have our own cannabis receptors, and we must have our own endogenous neurotransmitters. Why would we have a system? And then we do. We found this is one. There's several of them. If you just look at the overall 3D shape, it's pretty similar. And the receptors that work at are up here. And so again, these are on neurons. When the cannabis product or the normal neurotransmitter binds, what happens is this has a way of regulating this circuit. This cell gets excited. It releases this neurotransmitter. This could be a cell that's producing dopamine, acetylcholine, you name it. It could be any of them. But once the cell gets to a certain level of excitement, as measured by the calcium, it starts to create these products. They feed back to the receptors, and they modulate the synapse. They sort of have a negative effect. And so, again, what you're doing is this is normal regulatory mechanism. Our endocannabinoid system has turned out to be massive. It's a widespread system. Actually, if you ground up a brain right now and you measured how many receptors there are of all the different things I've talked about today, there's more cannabinoid receptors than any other one. And so they're obviously important in lots of different signaling processes. And however, we again are sort of usurping these by throwing in THC and other cannabis products. Marijuana contains obviously THC and cannabinoids in the old days. I've got to admit, not like Bill Clinton, I did try to inhale. Didn't really work. I didn't like the smoke. This is in the 70s. I just didn't like it. But pot back then had almost no THC in it. And now it's got huge amounts. Some have a 40% concentrate, way higher product seized in 2022 than even in 1995. So over 25 years or so, way more THC. And the effects of this THC are not always the same as our normal neurotransmitter system. This endocannabinoid system, I could show you, and I'm not going to because I'm almost out of time. It's got all these same things that happen with nicotine. You've got endocannabinoid receptors on all sorts of systems. They're on immune cells as well. They're all over the brain, involving all sorts of functions. Again, it's some of the same things you would see with nicotine, again, processing and focusing and so on. In adults, the transient changes are seen in memory and processing speed. So cannabis use amongst an adult, maybe not terrible. I mean, these effects are usually reversed, but not so sure in adolescents. In utero exposure has produced changes in speed and memory. This is in animals. There's some studies in humans. There's deficits seen in humans after abstinence, even after 6 weeks. So you'll have a 14, 16-year-old smoke some pot, do some cannabis product, and then they'll look at brain function, and these deficits persist. There's been some studies, again, to be intellectually honest, some studies don't support this, a lot of them do, changes in brain structure. Gray matter is basically the neurons. White matter is the tracks, the signaling pathways. And there's been one study that's been pretty good that even after low-level exposure in 14 to 16-year-olds, there's a loss of brain mass, loss of brain matter. So again, high potency, youth are using it. We don't know exactly about the endocannabinoid system, but I think you can guess if you disturb the signaling of the normal system, just like you did with nicotine, you're going to have some problems. Is it a gateway drug? When we were in, our youth, it was thought, well, it's not addictive. It's not a gateway drug. Well, those probably both statements are now false for compounds that contain THC. It is addictive for like 30% of the people who use it, and it's addictive in the same ways that nicotine and heroin are addictive. Okay? There's been, again, prenatal exposure in animals show memory learning tasks. Human longitudinal studies have been done now where they follow somebody for a number of years, lowered the IQ as an adult. Now, whatever you want to think of an IQ test, what's it actually measuring? But whatever it's measuring is changed in adolescent use, and it's changed in the long term. Adult smokers don't tend to use it. So again, maybe in adults it's not so bad. Again, I've got too much stuff on here. I'm going to sort of wrap up, but it's the same kind of idea. We have addiction, and when people stop, there's withdrawal. You cause brain changes that affect the addictive properties of other drugs, possibly. It has negative effects on a number of different functions in adolescents. And as far as we know, so far, the FDA has not approved the use of any product containing the whole cannabis plant, but it has approved the use of a couple different drugs. So I'm not saying that there's not. One of the issues with this field is that NIH and government agencies were very restrictive on allowing people to use THC products for research. It just wasn't out there for a long, long time until maybe the last 10 years. So a lot of the studies we just don't know yet. So I'm not saying that some of these things won't be great someday, but there's a lot of claims that are being made that just aren't supported by science yet. So we still have a possibility. There is an FDA-approved cannabidiol, CBD drug that's used for a certain couple sort of rare types of epilepsy. There's a couple drugs that are used to treat, probably several of you have even used this, Marinol, for treating cancer chemotherapy-induced nausea. There's some really bad nauseas. There's some pain that some THC products seem to be really actually effective for. So I'm not saying there's nothing there yet, but at the same time, we have problems that come about. And this is, again, partly because the cannabis products are having so much THC in them. And we say, well, what about CBD oils? Those don't have THC. We should be cool with that. Maybe. But some of the products that are labeled CBD oil or cannabis only, cannabidiol, they have THC in them anyway. And they don't always say that. They don't even say the right amount that's in there. And so people are finding that there's anxiety disorders, psychotic disorders, sleep disorders, withdrawal, and then a cannabis use disorder, which is very serious, really addiction. 16 million people have been diagnosed with this already. And so it's becoming, to me, one of these situations where people want to get high. I get that. Now you can buy a drug, used to be medical marijuana, now it's for everything. And many of the claims are just not supported yet, but they might be. For CBD, people claim it cures cancer, it affects AIDS. Nothing shows that. It affects COVID? No, it doesn't affect any of those. That's not been shown. Problem with CBD products is to me, a little more scary because it doesn't work through the same endogenous cannabinoid receptors very well. It does bind, but not very well. So the question is, where is it working? And I personally don't want to take a drug that I don't even know where the target is for the drug. Because it's sort of hard when you don't know what the target is, it's sort of hard to predict what the effects might be if you don't even know what it's going to. Again, there's some evidence that some of the cannabidiols or cannabinols are psychoactive in some way. Maybe that's because they've got a THC contaminant, but and so on. So the bottom line is, I've talked probably enough, but many claims of effectiveness are reported by some studies and not others. For pain, for example, for CBD, maybe a placebo effect, and maybe because they have THC contamination. So I'm all for lots and lots of research, But it's clearly many things it's claimed to be helping. CBD oil has not been shown to be the case. And CBD oil has been shown to produce changes in mood, sedation, and even some liver toxicity. So there is a therapeutic potential for this. And I'm not saying we shouldn't research it. I'm just saying that if you want to think of a drug affecting your brain, I sort of made this analogy to nicotine affecting an endogenous system, disturbing the signaling of it, disturbing development, maybe disturbing adolescent brain development. And I think you've got a parallel here too, where you have cannabinoid products disturbing development of a system that's very important, and we don't know how the system, our own endogenous system works, but we know that if we disturb it, there's evidence starting to be built that there are problems that come from this. And on the other side of the coin, there may be some therapeutic benefits we just don't know yet down the road. So more research needs to be done on those. So I'll stop yakking and see if you have any questions.
I have a comment. I have a number of patients who have leukemia, but they also, a number of them have had part or all of their lung removed for squamous cell. And in dealing with them, trying to understand how we can get them to stop smoking after they've already had major surgery, it's frequently impossible because of all the things you've just elucidated. And I have to admit, I've learned a lot today because it's not just a simple matter of having people who aren't very intelligent. There's a physiologic basis for this. And so one question I have is, I read where you do work with teenagers in the summer, and I was wondering how well they receive this information.
I give them a talk somewhat like this, and it's for the neuroscience department has a neuroscience summer camp, and they have around 60 or 70 students come in. They try to get students from all communities, all schools. We try to get a lot of underrepresented students coming in. We have scholarships for anybody who can't afford it. Then they have we've added on, got a grant that I was part of the writing of that. We got a grant to do like another 6 or 7 week research experience, sort of intense, but they do the week of neurocamp and then they do this. I think the students are pretty receptive. Again, these are smart kids. These are top kids, obviously. But I think they have some really good questions usually about it. And I don't ask them for a show of hands to see if they've ever used any of these products yet, but I hope, and it's the same way here. It's just, I think don't just say, people say, don't do drugs. Drugs are bad. No, that's because then people think you're lying, because when you do a drug, the first time is great. Like, my God, that heroin was great. The cocaine was great. The nicotine's great if you don't have and then people think, well, they're obviously lying to me. It's great. Well, it's great until it isn't. And so I think you explain the science to the kids, young adults, you can say, no, there's a reason why this is going to affect your brain. It may be okay now, but you see all these processes that it sets off and these complicated signaling pathways that get dysregulated. It's not going to be good forever. And I think that's the key to all this is education, is to really understand how these things work. And hopefully you'll be scared enough, or at least think, well, I'm going to really think about that, because now we know the mechanisms, some of these. And in the case of CBD, we don't even really know the mechanism of why these work. So I think they're receptive. I mean, that's the bottom line. And I don't know this, I don't know if it's a good idea or not, but I've thought about it. I have not applied for a grant, but I thought about trying to put something into this in a much simpler form and sort of some kind of PDF or something, and go on some campaign and find a way to get it distributed to a bunch of schools. I don't know if that's a good idea or not, but I've sort of thought about that, because don't just say don't do drugs, just say no. No, you gotta explain it, the science.
Thank you.
Yeah.
About how the half-life of nicotine is so much shorter in mice, and I was curious as why that was, and if that could be a way to somehow decrease the effects of.
Well, I think it's probably, I am hypothesizing here. It's because I think it's metabolized through different enzymes. And these enzymes that metabolize it are different versions in mice than in humans, so it gets metabolized very quickly that way. Rats is sort of in between. It's like 45 minutes or something like that. So the issue is some of these studies that are done on behaviors, there's always that caveat, and there's always caveat in any animal research, because we're not just a big rat or a big mouse or a big zebrafish, is that how do you administer it? And nicotine is always a very tricky field as to how to administer. You can put in like osmotic pumps to get a certain dose. You can have them self-administer. They can inject. They can inhale. And so the question is, are you comparing apples to apples all the time in these paradigms? And I think there's some good studies that sort that out, but that's one of the things they really have to consider too, there's some work done on primates, non-human primates, but again, as you know, people don't really like to work with them anymore. They're expensive, ethical concerns. So there's not a lot of it still done with other animals. Yeah, Dick? Oh, you're waiting for the mic. Okay.
Thanks, Tom. One of the important things that pharmacologists try to do is find drugs that will pass the blood-brain barrier.
Yeah.
And a lot of drugs that do have neurological effects, they can't get across the blood-brain barrier.
That's true.
But yet all of these drugs that you're describing do that.
Well, yeah, because in part,
is there anything to learn from that?
Well, I think it's mostly their chemistry. Like nicotine is a very lipid-soluble molecule. So it sort of goes everywhere. Since cells are lipid membranes, it goes into cells, it goes all over the place. And so that's sort of the issue is that one experiment I showed you, is it really working through the receptor, or is it just the fact that it just goes through the cell, gets in there, does something? And it may be both. But, the endocannabinoid products, again, are, I'm not a chemist, but if you see the structure of that, they're very lipid-soluble kinds of molecules too. So that's an easy problem. You bring up a good point, though, is trying to find drugs that treat this. That's always a problem in all of chemistry and pharmacology is, can you get it to the brain? This is a great drug. It works. It's fantastic, but we can't get past the blood-brain barrier. It's not like you think of it as it's a block here and nothing goes past. It's the vasculature has permeability to certain compounds selectively, and so you've got to design a compound that works and can get into the brain. And sometimes I've got a great compound, but it can't get in the brain. I got one that gets to the brain, but it doesn't work. So.
Where do the nicotine drugs that you take to stop smoking work?
Well, the one that's really had a little bit of work, but not much, it's still not that good, is Chantix or varenicline. And what it does is targets that one type I showed, the alpha-4 beta-2s, the high affinity ones. And what it does is what's called a partial agonist. And so what it does is it gets to the receptors before nicotine is the idea. It produces a little bit of an effect, but because it's already there occupying it, you don't really get the full effect of nicotine. And the idea is that you do a partial agonist, you blunt the effect, the brain starts to have some feedback. Like I'm not really, the brain is, the signaling is like, we're really not getting the full effect of this anymore. So people stop using it. Generally, when the brain doesn't get what it wants from a compound, it sort of stops using it. And so that's the way to do it. It still doesn't work. I think it's, I can't remember what percentage. It's a low percentage of people who use varenicline actually succeed. There's nicotine replacement therapy. You're still giving the drug that's bad, but at least it's better than the rest. I think one of the best things has got to be counseling. It's counseling, it's education, some drug support, to help withdrawal. But I think a lot of it is, from what I've seen, it's got to be interaction with people.
Why would nicotine replacement therapy work?
It just is a way of saying, I'm going to give you your drug, just like people do, other drugs of abuse, and they start lowering the dose down. So again, you're sort of saying, I'm getting my effect. I'm going to take less. Your brain's supposed to adapt to that and say, okay, I'm not really getting my effect anymore. And you're sort of weaning off. And part of that, you're weaning off so you don't get that withdrawal. Because nicotine withdrawal can be very serious, like withdrawal from other drugs of abuse. The sweating and the nausea and all the bad things that happen. So it's an idea of sort of weaning someone off of that. But since you know that nicotine does a lot of horrible things just by itself, You say, well, we're eliminating all those horrible carcinogens in the cigarette smoke, but the nicotine's is suppressing the immune system, it's promoting cancer growth, it's disturbing signaling just by itself. So it's better than nothing, but it's not great. But people are stopping. The number's going down. I think it's still education. And if you get people to stop, don't ever start. But the problem is, you see, once you start, it's really pretty hard to get off. And I'm afraid that's what's happening with cannabis products too. People are starting young, and it is addictive for 30% of the people, and then they have trouble getting off.
So could we have an endocannabinoid approach like we do with nicotine and Wellbutrin?
What do you mean? And
Prevent to
oh yeah, well,
to block it.
they may be working on that, but the cat's out of the bag. People love cannabinoid products, and it's partly because they just want to get high, and maybe they do have some therapeutic effect. But nicotine, you could say, does it really have any therapeutic effect at all? Not really. Where cannabinoids, you might say, well, there's some effect on nausea and pain, and there may be some good things that ultimately could come out of those products. I'm not sure anything good is coming out of nicotine ever. All right, thanks.